Welcome again, friends. We are talking about the different type of replication scheme of viruses. Now we have talked about different types of virus. If you remember, type 1, type 2, type 3, and these are the viruses. Remember, type 1 virus carry double-stranded DNA. Type 2 viruses carry single-stranded RNA, and it can be positive-stranded as well as negative-stranded. Now type 3 type of viruses carry double-stranded RNA. So we have seen all the replication processes of this kind of genetic material containing viruses. Now in this video we'll be talking about the type 4 and the last type of viruses, and these are called uh, the viruses which carries usually RNA or positive stranded RNA material. But so let me talk type 4. This type of viruses, they carry positive mRNA, no problem about that. So you may probably wonder that why shouldn't we put them in the category of type 2. Now you shouldn't put them because type 4 viruses, though they are having positive stranded mRNA, but they usually provide the replication scheme via a DNA intermediate. That's make this type 4 type of viruses completely different or unique. So let me talk type 4 viruses. So let me write it here. Though they are having positive stranded mRNA, but they replicate through a DNA intermediate. They replicate through DNA intermediate, and then they provide us the viral or virus particle or virion. So this is the scheme for the replication of this type 4 viruses. Now this type of viruses are also called and known as retroviruses. Retroviruses. Example for this virus is pretty common, and this is HIV AIDS. Remember, it is HIV virus causing the disease AIDS. So virus is HIV or human immunodeficiency virus, right? So this HIV virus, or actually HIV is the total form of the virus anyway. So now let's talk about the replication scheme of these viruses. Now for this purpose, what we are having, we'll be having a positive strand of virus, right? So usually we are having this positive strand. So let us talk about it. So we are having this positive stranded mRNA in the virus. So the virus will look like something like this. So if I draw the virus, it will look something like this. So let's say this is the virus, and inside the virus we are having this positive stranded mRNA. And after the unpacking of all these particles, the positive stranded mRNA will come onto the cytoplasm of the host cell. And as we are having this, so they use this positive stranded mRNA as a template, and what they can produce is a DNA segment, not the RNA. Remember, in previous case, what we have studied is that if we use positive mRNA as a template, we can produce negative mRNA. If we produce negative mRNA as a template, we can produce positive mRNA. Now, in this case, we will be seeing that we are using positive mRNA as a template, but we are producing a DNA strand onto the complementary of that, right? Now, how can we provide that? If you remember in the previous slides and previous discussion, we've always added here the enzyme which is required for this transcription, and the enzyme is called RNA polymerase, right, for the transcription. But in this case, it was RNA dependent RNA polymers in all the previous cases. But in this case, to produce a complementary DNA strand uh, with this positive stranded mRNA as a template, we need to provide DNA polymerase instead of RNA polymerase, right? So we should put here DNA polymerase. And obviously, we are using this positive stranded mRNA as a template, so this DNA polymerase will be RNA dependent DNA polymerase. It's very rare that we are having RNA dependent DNA polymerase, but we can have RNA dependent DNA polymerase. So we get RNA dependent DNA polymerase. Now if you use RNA dependent DNA polymerase here, and we use positive mRNA as a template, we can produce a DNA complementary to this positive mRNA there. So after this process, what we can produce, let's say here, So let's say this red thing is, let's say this one is a DNA. So what we can produce is this DNA strand, right? So we have the hybrid of RNA and DNA in this case, right? So this is a DNA RNA hybrid that is being generated here. Okay. Now, once we generate this DNA RNA hybrid, this can be integrated inside the host cell. But before, so sorry for that, but we cannot integrate DNA RNA hybrid anywhere, because if it is also having ribose sugar backbone, it is having deoxyribose sugar backbone, so it's a kind of unstable structure. In all the time, if you are getting this DNA RNA hybrid, that means it's very much unstable. So what we need to provide, either we need to provide both the strands with RNA or both the strands with DNA. Now in this case, what we'll be doing, we'll be providing both the strands with DNA. Why? Because the viruses, this retroviruses, require a DNA intermediate. Remember, here comes the DNA at the first part during this uh, process of replication. And after this stage, we'll be seeing that it will dissociate this RNA part, and it is using this DNA template as a template. So this DNA part as a template, and they are dissociating this RNA. So they won't require the RNA because they don't need this RNA anymore. So what they will be doing here, they are using this DNA strand as a template. They're using this DNA strand as a template. Now as they're using this DNA strand as a template, what they can produce here, so if this is the DNA, sorry, this one, if this is the DNA, again wrong one, sorry for picking wrong color, this is the right color, yeah. If this is the DNA strand previously, use this one as a template, what it can produce is another DNA strand. This light yellow color is the new DNA strand. So what they are doing in this case, they are dissociating this RNA strand out, right? So what we are providing, what they produce here is a dsDNA. So here this one is no longer a hybrid. So once we produce this double stranded DNA, it is ready to be incorporated to the host genome, because host genome is also made up with double stranded DNA. So once we produce this double stranded DNA by dissociating one RNA strand from this complex of RNA DNA, we can incorporate or insert this double stranded DNA inside the host genome. So if I draw the host genome, for example, let's say this is the host genome. Say, so let me change the color. Let's take this green. Okay, so let's say this green region is the host genome at the very beginning. So this is the host genome, double-stranded DNA. Somewhere there they are incorporating this DNA. So after the incorporation, what we can see is that, like that, they're having like that, and here will be this, and so you can see the incorporation is done at this page. So it is a integration of viral genome in viral DNA with the host DNA, viral DNA with host DNA, and it is getting integrated here somewhere here. Okay, so after this integration, they will start using all the proteins and enzymes that are being produced by the host cell, all the other protein synthesis machinery of the host cell, and they are guiding this process to provide or generate positive stranded RNA or positive mRNA, which can be easily generated from a double stranded DNA via a process called transcription. So they go through transcription using DNA dependent. So remember, here in this case they will be using DNA dependent RNA polymerase, right, for the transcription. You all know that, right? So they are using this RNA polymerase, and what they will produce this positive stranded RNA, so positive mRNA, right? And what we know, that this positive mRNA that they are providing here, this is this was the actual genetic component of our retrovirus, right? So we get our genetic component. Then what we need to have more, we need to have more proteins in this case, right? So we'll get proteins. So in this case also they are generating more and more mRNA, more and more products. Or from the other point of view, say they are producing more and more short RNA fragments. From these short RNA fragments, what they can generate, they can provide us structural proteins or capsid proteins. So these are the viral proteins, and a maximum of them are structural in nature. Structural proteins are generated. Once the structural proteins are generated, they will take both these structural proteins as well as positive mRNA, positive stranded mRNA, and they will cover this positive stranded mRNA inside this capsomere or capsid of the virus particle. So they will generate here and attach here like that, and what they produce is a matured progeny virus particle, right? That's how they can provide us the progeny virus particle there. So you can see that at the very beginning we started with this virus. Now after doing all these things, we are producing the progeny virus. But at the middle, what we have done, we have incorporated the DNA. The virus have incorporated the DNA into the host genome, and for that purpose they require to convert their positive stranded mRNA into double stranded DNA for the incorporation, right? So they provide this, and then the rest of the process, and finally they produce the structural proteins and engulf it, right? So that's how these retroviruses complete their replication cycle, and I hope that is helpful. Thank you.